Getting here we are. Stop over recording. Yay! All right, everybody. So, oh, I don't have the screen down. That's silly. Oh, right. It'll take a second. So I was just going to briefly review that last animation. There's one more that's on the cross bridge cycle, and then we pretty much have concluded this first PowerPoint sequence. And to some extent, it ties in a little bit about with today's lab. Today we're in the zoo lab. I'm sure you got the email. And so I've got it more or less set up. We just have to pull out some dissecting microscopes and some regular microscopes. I have the stuff out by and large because most of it's in the freezer I have to remove. And then I sent you the PDF file for the handout. So that's where we are. So everybody can see what's going on there. Voila. Oh, thank you. Just right. Difficult. So I again, you don't necessarily have to. I'm not going to play it with the sound or anything like that. I just wanted you to start to see this come of age here. So remember, this is just basically the concept of transitioning the excitation contraction coupling. So where this starts is what's going on distinctly within. And they're showing you basically a series of sections of within a muscle fiber, the cell itself, and then we'll look specifically within the sarcomere. And again, this is a very good representation to me of transverse tubules. You could see it's not they penetrate and surround each. Let's go back and start again. So I tried to stop that in the middle again. I just did. Hard to do with these. As you can see, it surrounds each individual fibril. So what they're encircling, but they're effectively within the entire structure. So it's a very interesting and unique way to generate this signal throughout the fibers. And roughly it is at the A and I band junction. So they're giving you a nice little panoramic view so you can see it. And now we're looking at the sarcoplasmic reticulum, and you can see the distinction between that and the T tubules by color. So here you can see the transverse tubule. That's the excitation. In order for all of this to occur, we have to reach an action potential. We have to reach threshold. Once it reaches threshold, that signal will move sufficiently along the sarcolemma, along the cell's membrane itself, past the area where the contact points are, where the motor unit is, and trigger the start first voltage-gated sodium and then voltage-gated calcium. First voltage-gated sodium and then voltage-gated potassium channels. Once that occurs, the sodium will rush in, that'll depolarize, potassium rush out or repolarize, and basically it's a, for lack of a better way to describe it, just like we do in nerves, it's a wave. Rapidly changing the polarity along the membrane, and there's just a series then of channels, of voltage-gated channels. And what they do is they will send that signal, as you'll see depicted here, down along here into the membrane, and on either side of it, sort of in that bluish color, are the terminal cisternae. Cistern means a well. They're filled with calcium. It's intracellular. The voltage-gated channels will trigger voltage-gated channels as well in the cisternae. So basically, that wave will unlock the ability for the calcium to flood the interior of the cell, bond, as you're going to see shortly, bond with the troponin, allow the tropomyosin to move, and voila, we have contraction. That's the coupling. There are the voltage-gated areas that you hear, the voltage-gated proteins. If you want to just sort of generically, you can see one aspect of it on the T tubule and one on the cistern. And voila, there goes the calcium. And what transpires? Contraction. Basically a shortening and overlapping of the thick and thin filaments, and we're going to see that again here. And that's everything. So a quick, and I do like to do that, as you know, taking and looking at what happens. So what's the next step? When the calcium's back in the well, the tropomyosin is in its proper position. It's held by the troponin. It's blocking the myosin binding sites on the actin and the actin binding sites on the myosin. The fiber remains relaxed. Once the calcium floods, higher intracellular calcium binds to the troponin. And again, anything that binds to a protein changes the protein. This is all about shape. So it will then allow the tropomyosin to slip away, expose the binding sites, cross-bridging, as you'll see shortly, as long as we have calcium in the area, and which will be in the area as long as there's signal being produced, it will continue to contract until it's maximally contracted. And then when it ceases, that will be the signal to pump the, and again, this requires, there's a lot of energy in muscles. So you're going to see we have energy to load it. We have energy to create an action potential. We have energy that effectively, as you'll see, resets the muscle. When we go back to this quote unquote resting point, even though the myosin heads are in there, are ready to fire, and we need some more energy to pump it back in sarcoplasmic reticulum. So there's a lot of energy expenditure associated with muscle and also with nerve activity. Like right now, hopefully your brains are more fully engaged, which was not the case with Campbell, the Detroit's coach.
17 points that they had to leave
now. How can you do? And the other one is the guy Flowers from Baltimore, another brilliant effort. God, talk about composure and discipline on a team. That's never been a strong point of that particular group. You know, the coach he's always the smartest guy in the room. Never been a hardball who wasn't the smartest guy in the room. Moving on. So what that brings us to is actually how the contraction occurs. What causes the filaments to slide? There are 4 major steps. It starts with the fact, and it has to start with the fact that we have energy effectively resets the cycle. So when they talk about a high energy myosin head, it's gone from its contracted position to its cocked and loaded position, so to speak. So you would like a trigger on a weapon. What do I know about weapons? You can fit thousands of them in that can. Okay. When the heads come in contact, as you will see specifically how it generates the quote power stroke, this basically, so it's a motor protein. It changes shape with the input of energy and the attachment of these two structures, or with the attachment of these two structures, since the energy is already preloaded. Effectively, the head moves, or they describe it as pivot, and the thin filaments are being pulled toward the midline, and so, because they're attached to the Z disc, pulling them closer together. Then what requires additional ATP to the myosin head to cause detachment. So it's got to, cross bridge has to form with the input of energy. We have to generate a power stroke. Then to complete it, whether it continues contracting or we're done, we have to have ATP necessary. That's really important. Cross bridges to detach. And then we have to effectively recock the head. And that comes from the hydrolysis of ATP going to ADP. So ATP attachment in and of itself will allow for the detachment of the myosin head and the active site on the actin. But to reset it for the next cycle, good to go, you have to hydrolyze that ATP. And you'll see it. And they'll show you this graphically. I think the animation does it better. And it's very hard to read. That's why I don't like this. But what are you going to do? But let's see if we can hear this relatively well. I had an animation in class earlier for micro, so I do want to get the volume level set.
The contraction of the skeletal muscle generates the force necessary to move the skeleton. A contraction is triggered by a series of molecular events known as the cross-bridge cycle. In a skeletal muscle fiber, the functional unit of contraction is called the sarcomer. A sarcomere shortens when myosin heads in thick myofilaments form cross bridges with actin molecules in thin myofilaments. The formation of a cross bridge is initiated when calcium ions released from the sarcoplasmic reticulum bind to troponin. This binding causes troponin to change shape. Tropomyosin moves away from the myosin-binding sites on actin, allowing the myosin head to bind actin and form a cross bridge. Also note that the myosin head must be activated before a cross bridge cycle can begin. This occurs when ATP binds to the myosin head and is hydrolyzed to ADP and inorganic phosphate. The energy liberated from the hydrolysis of ATP activates the myosin head, forcing it into the cocked position. A cross-bridge cycle may be divided into 4 steps. Step 1, cross-bridge formation. The activated myosin head binds to actin, forming a cross bridge. Inorganic phosphate is released, and the bond between myosin and actin becomes stronger. Step 2, the power stroke. ADP is released, and the activated myosin head pivots, sliding the thin myofilament toward the center of the sarcomere. Step 3, cross-bridge detachment. When another ATP binds to the myosin head, the link between the myosin head and actin weakens, and the myosin head detaches. Step 4, reactivation of the myosin head. ATP is hydrolyzed to ADP and inorganic phosphate. The energy released during hydrolysis reactivates the myosin head, returning it to the cocked position.
It's a very good
As long as the
illustration.
binding sites on actin remain exposed, the cross-bridge cycle will repeat.
And it will do so
And
to
as
its
the cycle
physiological
repeats,
limit.
the
When it gets-
thin myofilaments are pulled toward each other, and the sarcomere shortens. This shortening causes the whole muscle to contract. Cross-bridge cycling ends when calcium ions are actively transported back into the sarcoplasmic reticulum. Troponin returns to its original shape, allowing tropomyosin to glide over and cover the myosin-binding site on actin.
So effectively everything, and it's very important, is preloaded. As you see here, as they conclude this, and it's good. So, I mean, it just underscores the magic of ATP. A molecule we're constantly taking apart and then constantly remaking. So it's extraordinary, and I'm sure I've told it to you. I mean, if you weigh 150 pounds, you probably generate and break down 150 pounds worth of ATP every day. Amazing amounts of this extraordinarily versatile molecule. So any way you want to look at it. And here you can see that head, when it's ready to go, has the ADP and the phosphate. When the bond occurs, the phosphate releases that, tightens it. Then the ADP releases that, pivots it, and then the next ATP comes to the party, and we're set to go all over again. Love it. A lot of expenditure of energy, a lot of investment of energy, comparatively speaking. So it's always interesting to me how we do so many things that really don't fatigue us. As long as we're regenerating, it's like taking a nice walk. You know, it's effectively, it's an aerobic activity depending on how much your heart rate, if you're on treadmill. And even somebody who's out of shape, you can do it for a very long period of time. My wife informs me I had better start doing it again. Which brings this, and I love this. See, years ago, forensics became very interesting. It was pre the advent of CSI. Okay, the crime show that has been made 6,000 times at every location known to man. It's like NCIS, same deal. And what happened was we knew about this, okay? But people really wasn't a tremendous interest in it A lot of it, interestingly, was stimulated by the O.J. Simpson trial. When we started to look at DNA evidence and forensic evidence and collect it. Then you had, this was 30 years ago. It's fascinating, a little bit of history. But rigor mortis is something that pathologists and crime scene people utilize to estimate time, because time of death gives you the window of when a perpetrator, assuming it was a criminal act, could do something. And so what happens is muscles begin to stiffen 3 to 4 hours after death, and it varies with atmospheric temperature and the temperature of the body and other conditions that are there. And peak rigidity, which you can determine by feel, 12 hours post-mortem. So the intracellular calcium levels come first. So remember, person's not living, so there's no ATP. The calcium can't be replaced in the sarcoplasmic reticulum. So the integrity of the cisternae in the sarcoplasmic reticulum is what goes first, and it allows the calcium to flood. Now that's the rigidity. And then after X amount of time, again, depending on atmospheric conditions and local conditions, then now it doesn't relax until the muscle starts to deteriorate. The actual proteins themselves, like roadkill. And you see if, the legs are up or whatever, that type of thing. So you're going to get cross bridging. So results, the myosin head stays bound, constant state of contraction until we get denaturing of those proteins. Interestingly, there's a similar thing that goes on in the bloodstream if you're not aware of it. It's called liver mortis. So we look at rigor. Liver is when, that body was moved. So when they examine a body that's on its belly someplace, and they look on the back and they see what appears to be blood incorporated with the skin, that meant that person died and was on their back when they died for whatever reason, nefarious or otherwise. And they were moved and tossed someplace out of a car or in the woods on their stomach, because it's going to reveal the position when they died. That's another one. There's all these neat forensic things. There was so much interest in this 30. I had just started teaching actively in this about 1998, and then all the schools were talking about forensics and getting a forensic department. There's so much interest in it. Not a lot of that. Duquesne's always had a good name. That's because one of the most famous pathologists in the world at the time, and to some extent for a very long career and very active politically, is Cyril Weck, who was the county coroner here. And it was his deputy that was the whole concussion thing. So there was a lot of, so he was like, they actually fashioned a show, Quincy, which is a very successful show in the 70s and 80s with late Jack Klugman, about him, who had occurred in LA, because there was a similar character in the LA coroner's office. He was like the deputy coroner for the county. Fascinating. Cyril Weck was the head of our staff at where the arena is now, where PPG Paints Arena is, because we had a hospital there. That was the old Central Medical, later St. Francis Central, and he was the head of the medical staff. So a little side note there. You know me, always a side note. We're moving next. See, they even got St. Francis Central. Which brings us, thank you, I know I'm presenting to everybody. You don't have to tell me twice. Voila. Now we're going to get a little bit more into the physiological factors of energy utilization and contraction. That's how it happens. So whether it's a single or a whole muscle, and it's all about tension. This is a distinction most of you are aware already. I would think about isometric versus isotonic contractions. We start more or less isometrically until once the muscle begins. So isometric, iso for same, metric for length. So when you start to lift something, until you accumulate enough activity of muscles, depending on the weight or the mass of the object, at that point, you're just activating isometrically. Once you start to move it, now it goes into isotonic. The tension remains the same, but you're shortening it. And so, and exercises have always been a combination of those. Isometrics, people could do in limited spaces, pushing on walls, because you're pushing effectively on an immovable object. You're just spinning your wheels, so to speak, but it doesn't mean you're not generating muscle activity. And then for years, then there was a combination of doing them both. It was called isokinetic. And really, it was a fine word for saying that you had a device that you'd maximum contract and it would be released very slowly. So you could basically have the benefits of both isometric and isotonic contraction. That came about 60, 70 years ago when they began to, so the exercise aspects of it understood that are there. So it varies. So every muscle, even the tiniest ones, have at least one motor nerve, but a lot of axons and a lot of terminals, each of which we call a neuromuscular junction. So the motor unit is really the neuromuscular junction, if you want to look at it. When we're looking at a particular axon with as many areas as it goes to. And so some have very limited, some have, and it kind of has a lot to do with the function. So it consists of the motor neuron and all of the fibers. So it can be very few, okay? So if it's just a couple of isolated fibers, think of your fingers, the digital control we have with fine activities, knitting, sewing, whatever type of manipulations you have, fine motor control. They're spread, and they're not all in one area. They spread throughout the muscle, so it's only a limited contraction, and to some extent that avoids fatigue. So it's very well arranged in that regard. So even when you're trying to lift that heavy object, as you're bringing more motor units into play, they're not all down here, they're not all up here. They're spread throughout. And it's just, again, I think it's very well understood. So here we are. So you can see a nerve here coming in, and another nerve here coming in, going to different fibers along whatever muscle that happens to be. And there's the micrograph of it. So you can see how they got the animations, how you can see how the axon, the little bulb, rather than pointing in, kind of lays a little flat, as you saw on the previous animations. You know, you're trying to, when they create these graphics, they take them from that. And it really has been a vast improvement over the years, I think. This is something we're not going to do this week, but next week we're going to do a little EMG stuff. Depending on where we are with the nervous system, we might do a little bit of that stuff as well in lab, because we've got a whole week to listen to me spout off about this stuff. Blab, verbal diarrhea, whatever you want to call it. So the twitch, okay, a single action potential from a motor nerve, very rapid, not discernible. And twitches are not something that happens normally. If you see someone who has a twitch, normally that's an abnormality in the nerve or in the system itself. I mean, I can see the response when my wife has one of those jolts from her trigeminal neuralgia. I mean, I can tell. It's really not a nerve. I know that there's a signal being sent. And it results sometimes in a twitch. But when someone's, twitchy, as it were, normally there's an abnormality. And originally we refer to these as EMG, electromyographic studies. They've been around forever. Electrodiagnostics and electromyography have been around for a very long time. We can do it now. And years ago, they called it the needle test. You get somebody with a bad back or carpal tunnel, and you'd stick sterile needles into the thenar eminence, the muscle here, and an electrode here with a stimulator, and boom, and you'd be able to observe, listen to, because the sounds also can sometimes reflect problems, and you'd observe it on an oscilloscope. That was the way you looked at the tracing, or sometimes with a stylus as the paper moved, which was the classic way to do both EEGs, EMGs, ECGs. So all those studies, we moved the paper. Now it's a little bit different now with the technology we have, but it was the old days. It was an oscilloscope, which you should know is the precursor, or really sort of a television set, very monochromatic. Yes, sir.
Where does the neuron originate in the brain?
In the motor cortex. So my motor telling my thumb to do this was coming from here. Remember, we looked at that homunculus in chapter 12, the central nervous system, and it had like a map of what part of the brain. And when they talk about the motor cortex, that's also known as the precentral gyrus. So remember, this was the central sulcus. Precentral was where the actual motor signal went to the spinal cord or cranial nerve, whatever it was. And then you have the postcentral, which is the sensory aspect. Okay? So And so we look at these tracings, and I'll show them to you. Things that we looked at to see if there was a nerve impingement, because the numbers are known depending on where you put the stimulator and where the receptor is over here, where the pin. And now we just use, like the pads we use for, which I just ordered more of. So the events where there's always a tiny gap between stimulus for excitation, contraction, coupled to occur, microseconds. Okay, we don't see it, any tension. Then we see the contraction, and then we see the relaxation associated with the calcium entering and cross bridging and then leaving. So, and it always contracts faster than it relaxes. That's a fine point. So you see the curve. So here's the stimulus. You can see the latent period, 5 milliseconds, whatever they have here. The contraction to maximum contraction, 25 milliseconds, 1/40th of a second. And then slowly, and here within 1/10 of a second, more or less, you're back down to complete relaxation. So we can do this on an electromyogram. We don't see them in real life that often. Why is this important down the road? Is we utilize what we see here to try to produce things in rehabilitation. For you folks in sport, I'm sure they do electrical stim for your, there's a whole science, and it's nothing new on hip brain. The guy who taught us physical therapy in 1973 was my physical therapy director at the hospital I residency at, 77 to 79, wrote articles in 1936. So even then, he was probably in his latter 60s when I knew him. So they were using, and our orthopedic surgeons were really good using an electrical stim. They had the rep of being the best to it probably in the 50s and 60s in Philadelphia. Okay. So duration and strength of the twitches play a lot of roles. So we have muscles that are rapid, blinking your eyes, and we have muscles that contract more slowly. And there's a lot between the way they contract and the nature of the fibers that determine whether they're postural muscles, power muscles, different ways that we use them. So this is a perfectly good example. Think how rapidly you can move your eyes from point A to point B, looking lateral rectus moving to the outside like this and that, okay? Very rapid. Boom, boom. Gastrocnemius is the big muscle, and you have 3 muscles in your case. 2 heads of the gastrocnemius, 1 giant soleus. Their conjoined tendons basically spiral around each other and make the Achilles tendon. Okay, the one that you're constantly using when you're standing is the soleus. It's a postural muscle. Every time you do a little changing position, it's pulling your tibia into the right position. If you're jumping up to get a rebound, my half-inch vertical leap and all that, as you're well aware, okay, I don't go fast for anything unless it's food. So I'm back on diet. Back to keto. So much cheese, so little time. It's all right. You may not believe it, but because you guys weren't even here on camera, I was down to 180. I gotta drop out 30 of these pounditos. My numbers weren't that bad. The doctor wasn't giving me cholesterol medicine or anything like that. He did advise me to be a little more careful. My wife yelled at me. But if you're jumping, the gastrocnemius is what lets Michael Jordan go grab a rebound. Okay, so it's relatively smooth, twitch laboratory created or PT created, and typically unless someone has a neuromuscular problem of some types. And there's different kinds of responses depending on demand. And so we can evaluate this by changing the frequency and changing the strength of the stimulation. Frequency is a bigger player. Again, when you're dealing with nervous system, frequency is the key. It's not how big, how many. How big plays a little bit more of a role in muscle. With nerve, it does not. So isolated stimulus, isolated twitch. Contracts, relaxes, contracts, relax. We just see this. So here you're looking at the signals, let's say every 100, every 10th of a second. Okay, that type of area, somewhere between a tenth, 150 milliseconds. So you completely relax, is isolated. Now, what happens, and it doesn't happen with nerve so much, with muscle, because before it completely relaxes, if you re-stimulate it, you're just adding to calcium that hasn't been pumped back in to the sarcoplasmic reticulum. So if you add additional stimuli comparatively rapidly, and they can't completely relax, we end up with more calcium. And so this is classically referred to as a staircase pattern. It's just like up, 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 like you're climbing a set of stairs. Notice the frequency now is what? Maybe every 50 milliseconds, maybe 20 per second. Okay, rather than one before was, let's say, 10 per second. But we're not, so we call that temporal over time because we're increasing the number of stimuli over a unit of time. Now, if the stimuli increases yet again near maximum, smooth, continuous contractions, that, and the word we use is summate. So at that point, if we continue to make them faster, okay, we can progress the muscle to a sustained quivering referred to as unfused or incomplete. Now, unfused implies quivering or a little bit. It's contracted, but it's not just not moving at all. They're still, you can almost palpate or feel some motion. And that's where the disease tetanus comes from, permanent contractures. And this is unfused, so electromyographically, you can see it. The stimuli now are what? 1, 2, 3, 30 per second, let's say. 3 in a 10 and 100 millisecond interval. 30. Okay. So that's unfused tetanus. And we're not done yet because we have complete tetanus. So if we increase them further, and this is all done in the laboratory or in rehab, we will, because contraction will fuse one smooth contraction. And then if we maintain it, it will lead to muscle fatigue. So now you're seeing maybe 100, 75, 80, 100 stimuli every second, every 10 milliseconds or so. Why will we do this? If someone comes in with a spasm you can't break that's not related to a disease entity or something like that, we used to do it for back spasms all the time. You'd induce tetany, okay? Or if you wanted to work the muscle, you could do it reciprocally. You could move it one way and the other way, depending on the pads that you put on. And you had a device called a low volt stimulator because you're not damaging anything. Or you could use surge to try to increase muscle strength if the person was unable to move. So a lot of this is done physical therapy wise. And we've had a lot of students here who have gone into doctor of physical therapy programs and have done very, very well with it, who came through the same kind of background you do. So I've got a fair amount of experience with this because most of my surgery was orthopedically oriented, a lot of people with sprains and strains, ankles and knees and things like that. So what's happening is you're recruiting more muscles leading to more precise control. So you can either not get subthreshold, nothing happened, threshold, observable contraction, maximum, strongest contraction. It's all about how many motor units. And this is the nugget that has to do with how powerful your stimulus is. So if you're subthreshold, nothing's happening. If you get to threshold, isolated motor units, even if you increase it. Once you get to maximum, what does it mean? All the motor units are excited. So even if you go over maximum, you're no more benefit. You're going to get the same amount of contraction. That's why the temporal summation and the increase, why we use that therapeutically, max isn't really that terrible. Once you get to max, then you just do it more rapid. So, changes in stimulus strength, size principle, motor units smallest first, larger next, largest last. Okay, asynchronously, so it doesn't fatigue, so they're not all contracting at the same time. That's part of it. Well, and you can see smallest, medium, largest over the course of time. Muscles are always, to some extent, even when you're asleep, there's still a certain amount we call muscle tone, known as resting muscle tone. In surgery, you have to take that into account. So when someone's anesthetized, there's not a whole lot of tone. You have to have an idea. Let's say you're doing bone and joint or tendon muscle surgery. I mean, you could easily end up, if you're releasing a finger contracture, they might wake up and the finger's all of a sudden like this, and there's no way to get it back. I mean, you have to take, so if you overcorrect it, to know what you're doing. So, and that's really a tincture of experience. To understand what the appropriate resting tension is, pre-op, post-operatively, etc. I mean, there's a lot of nuances. It's not quite the same as somebody's got a lump, you got to take it out. When you're trying to reposition things, how common is ACL grafting? Hey, it's easy to mess those up. So alternatively activated in response to stretch receptors. So they're always there. If someone's comatose, then we begin to see that. And again, with muscles, what happens when they're not being used at all? Atrophy. So even people in coma, okay, we're having physical therapy and bedside services, exercise them in the hopes that if they do come back, that at least we have, because if a muscle atrophy is too much, it's not coming back. It's not the same as you having a cast or a brace on for 3 weeks and feel, and a couple of weeks out of it, all of a sudden, hey, I'm fine. That's the beauty of being young and healthy. Something I no longer have. Where's the brush here? Isotonic, same tension, changes length, and you can do so concentrically or eccentrically. What does that mean? Most time we think of contractions, we think concentrically. I'm firing my muscles, I'm bringing it closer. Okay. Eccentrically, I put it down, I mean, unless you want to break your phone or spill your drink, if you plop it down real fast, it's going to be all over the place. You're going to damage something. So you're using your muscles. Eccentrically means you're basically stretching the muscle or not. You're reversing the contraction, but still using a certain amount of force. So muscle lengthens with force. Concentric, a bit more of the way it works. So laying a book down versus picking a book up, for those who still use books. I had a whole thing with booksellers. So voila to that, and then resistance, not much there. Isometrics. So the same, and that's why I got, spinning their wheels. Basically, it's very, very popular exercises. Now people, and I've been out of the PT and trainers loop for a long time. I used to work with them. Maybe my uncle worked with the head trainer of the Steelers during his very, very closest to friends, best friends. So he learned a lot from the trainers for the big pro teams and the big college teams. And so he used to speak at conferences. He made me seem quiet. He had a gift for, how can I say it, Doug? BS. Great anecdote. We had a major conference every year in Hershey at the medical school. Everybody was there. It was a great conference. Appointment television. So I see my uncle, and they have a list. They give you a little pamphlet when you go to a conference of who's speaking and their CV, their resume. And I'm seeing, Edwin Stein, expertise in athletic training, founder, owner of Stein's Foot Specialties, which is our family's business, blah, 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 blah. BS, Princeton University. Because everybody else on there has a BS from here, and an MS from here, an MD here, a DPM here, and a PhD here. And he was insecure. I went to my dad. I gave my dad one of those, Dad, what the F is your brother doing? The only BS he's got. Because he says, I know, I told him. When he was in the Army in World War II, in 1945, Princeton University offered classes to the GIs. He was stationed in Germany. He went right at the end of the war, so he didn't see combat. 45. But he took enough courses, they figured he'd throw it in because he's got some credits from Princeton. He said, I'll just tell him it's a BS. And it was. That's an interesting side note for off-topic discussion. Understand wherever you're going to go and work, particularly professionally, they're going to look hard at all. Don't ever put anything on your resume that's not true. You'll be out the door. If you messed up someplace, If you had a DUI and you haven't had one for 3, put it in there. Because these days, these background checks, they will find it. Trust me. It's nobody likes surprises, not that kind. Okay? It's not like, oh, surprise birthday party. No. You know, particularly in the workplace, be really accurate. That will crush you. If you haven't heard that before, you can't hear it enough. How do we get energy? Well, we eat carbohydrates. So the energy needed moves and detaches cross bridges, blah, 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 sodium in, potassium out. There's a lot of energy. Okay. The ATP we have on hand within the muscle, barely 5 seconds. That's why nearly all activities start anaerobically, meaning they're either using up energy sources that are on hand or alternative energy sources. The only source of energy for contractile activity must be regenerated quickly. So and most of the time, that's all we use a muscle for. We reach here, we go here, we walk from here to here. A couple of seconds, you know, we've used up the available energy. How do we regenerate? We have availability of creatine phosphate. So, and this is the error. A lot, people were gobbling protein powders with creatine like crazy. Guys who are working out, gals who are working out. The reality is, look, it's a protein. Any protein would do. Because what is your body? You all know how your body metabolizes protein. It breaks down to amino acids. So you're making more amino acids. It doesn't matter that it's not like magically, creatine's going to magically, creatine's magically going to go and make creatine phosphate in your muscle. It doesn't work that way. Okay? If anything, remember, it's highly nitrogenated. And you've got to be careful doing that if there's any kidney problem, if you're not hydrating sufficient. So, but that'll help you. Not for long. And then at that point, you're out. Now you're in pure anaerobic activity. You're running a sprint. You're doing a 200. It takes, what, 22, 25 seconds at a very, very elite level. Okay. You're already into anaerobic by time. And then once you've used that up, now you have to either convert to aerobic. Aerobic is a very simple definition to me. You're just matching the activity with your respiration and your heart rate. Increase your heart rate, you increase your respiration accordingly. It's an endurance activity, and you can work on that. So the direct phosphorylation of ADP by creatine phosphate, another high energy phosphate, unique molecule in the fibers, can donate a phosphate and recreate ATP. Kinase is the enzyme. They have enough ATP and CP reserves. Another 15 seconds. Now we're at 20 seconds. So voila to that. At that point, it's anaerobic. Unless you alter the activity, you sprint to start, now you reduce your activity level, you're on the treadmill, you do X amount of miles in a sprint, now you go back to walking, replenish, etc. Generated by breaking down and using energy stored in glucose, glycolysis. You all know about that. First step in glucose breakdown, no oxygen required. Why is it anaerobic? Because you don't have any oxygen. Remember, the muscles are contracting. The oxygen can't get back in there. Those NADHs with the electrons, I know where to put my electrons. So what do? You put it back on pyruvate, it makes lactate, you got the picture. So only get 2 ATPs because you don't enter the Krebs cycle, whatever you want to call it, citric acid cycle, doesn't matter to me. Low oxygen levels prevent pyruvic acid from going into aerobic respiration. That's the ticket to get into the mitochondria. So what happens with the oxygen available and the bulging muscles playing a significant role when they're contracting, so it becomes anaerobic glycolysis and we get lactic acid. It's not all. The lactic acid, and along with weight, because there's no circulation, you have an extensive amount of waste products, electrolytes and other materials. And that's what causes people to spasm and cramp up and cause them to stop, as well as the oxygen debt. Your liver will be able to reconvert the lactate back to pyruvate, and you can use it appropriately once you stop the exercise. So lactic acid in the bloodstream still has a fuel potential, but the liver, once that activity stops, will convert it. So it's very inefficient. 5% as much ATP as aerobic, but very rapid. So that's why, again, in athletics, we can train people for either. If they're doing the short burst anaerobic activities, sprinters, if you will. Think of a tennis player, they're well conditioned. Tennis is mostly anaerobic. Stop. I'm still amazed that Novak lost semis. If you're any of you tennis fans. No tennis fans here. Oh, God. There's more to life than football. So at that point, aerobic respiration has to take over. 95% of ATP during rest and light to moderate exercise. Not as rapidly, it doesn't have to be. We all know about the ATPs. So fuels used include glucose from glycogen stored in muscle fiber, then blood-borne, then free fatty acids. So if you're exercising and you're on a weight loss, when you're doing a fat burn, once you're in that, after that first 30 minutes, now if you're trying to burn fat, it's that 30 to 40 minute area where all of a sudden it's like the keto diet. You're limiting carbs, so your body's, what's the next alternative? Use fats. And if it works properly, then the fats start to go even before you lose the weight. So all of a sudden that shirt doesn't look as tight, or that belt gets an extra. Oh, please. I don't appreciate that little clever smile. How you knew I was talking to you?
It wasn't for you. Don't worry.
Okay. I know.
Don't worry.
And so aerobic endurance is something that can continue even for people who are untrained for a long period of time, and anaerobic not. This summarizes it. Okay. And the whole idea of the fatigue nugget, as it were. Okay. Ionic imbalances, more so than the lactic acid. It's the accumulation of electrolytes, okay, disrupting membrane potentials, increased inorganic phosphate, interfering with calcium release. There's a lot to it. That's kind of, and as you get older, you're much more prone to spasms and what we call Charlie horses, cramps at night in your legs. I had one last night. And I take electrolytes, I take supplements when I do this diet. There's more of it. So increased magnesium, decreased ATP, magnesium levels increase, intervening with, as you can see, all those things are parts of it. So it's not so much the lack of ATP, more so waste products, I think would be a fair way. And to me, we used to call this an oxygen debt. Somebody liked to use the term EPOC. I don't know why. Replenish, convert, and everything reverses. Restore again, it's skeletal muscle. It's just like your liver. When you use the glycogen up in an emergency, fight or flight, you put it back in. Again, it's the crash cart analogy that I talked to you about. And then you resynthesize that, and that's everything. So we'll call it quits here. Got a lot done, a lot of material. The next one deals a bit with some more physiology, and then we look a little bit at cardiac. We do that at the heart more so, and then smooth, which is a very different muscle altogether. But it's smooth. Goodbye out there in virtual land.